Hello, my name is Nick Buttermore. Here's my student ID to confirm that I am me. And without further ado, let's get it on and talk about MHC class 1 and MHC class 2 proteins. So we're going to start with MHC class 2. So MHC class 2 proteins are utilized by cells known as professional antigen presenting cells. These can be anything from dendritic cells, macrophages, really anything that phagocytizes something, even B cells. And what happens here is these cells phagocytize exogenous antigen, meaning it's antigen that's not already within the cell, brings it into a vesicle, and from there within the vesicle, it is digested down into being smaller bits of the antigen. And from there, within the vesicle, MHC class 2 protein, which is derived from the DNA within the nucleus of these cells, will bind it to the partially digested antigen, and then ultimately it gets set onto the surface of these professional antigen presenting cells, where it will be bound to T helper cells that utilize CD4 co-receptor and their TCRs to bind to the MHC class 2, and then it results in a cascade of responses. And then on the other side here we have MHC class 1 proteins. These are characterized as being cytosolic processing, meaning that it's processed within the cytosol rather than the vacuoles. And this is what happens is you have endogenous floating antigen within the cell, and it gets processed by proteasomes, which are used for just partially digesting these antigens to being smaller bits, to where it goes into the endoplasmic reticulum and it gets bound to MHC class 1 proteins, which again are derived from the DNA of the nucleus. It gets processed in the Golgi apparatus, and then it gets presented on the surface of MHC class 1 cells. So what these types of cells are they're really any cell that's been infected by the antigen itself. That's why it's endogenous antigen and cytosolic processing. So when the MHC class 1 protein is on the surface of the cell, T cytotoxic cells come, bind to it with their TCRs utilizing CD8 co-receptor, and the T cytotoxic cells will lyse the antigen presenting cells here in the case of class 1. So major differences, again, to highlight these, MHC class 2 proteins are endocytic processing within vacuoles, and it grabs exogenous antigen. They're professional presenting cells that are not actually infected by the antigen itself, and they present to T helper cells, which utilize CD4 as a co-receptor. On the flip side, MHC class 1 proteins are cytosolic processing, which grabs endogenous flowing antigens, and it goes to class 1 MHC protein, and it presents to T cytotoxic cells, which kill the cell. About the structure, MHC class 1 has 3 alpha subunits and 1 beta subunit, and MHC class 2 has 2 alpha subunits and 2 beta subunits. Besides that, that's about it. So thanks for listening.